this on? Okay.
Yes.
Great, thanks. So my name is Adam Shawitz. I'm a physician working in rare disease drug development. Been working in rare diseases for close to 6 years now. And wanted to thank Christine for inviting me and Vanessa and Ashley down to speak about natural history studies and registries. And before I pass it along to Ashley and to Vanessa, I just wanted to give a brief overview of sort of what natural history studies and registries are and why we think that they're so important. And then we can go into Vanessa and Ashley's discussions. So this is a definition of a registry. This was taken from a government website, the Agency for Healthcare Research and Quality. One of the only places I found that was a real definition of a registry. And basically, it's an organized system that uses observational study methods to collect uniform data to evaluate specified outcomes for a population defined by a particular disease condition or exposure, and that serves one or more predetermined scientific, clinical, or policy purposes. So the key points are that a registry is an organized system. It's an observational study, or it's an observational collection of data. It's not a trial of a drug. It's not an interventional study. And it tracks specified particular outcomes, things that someone prospectively says, we want to look at this, we want to look at that. And it's focused on a particular population. So these are fairly broad, but you know the four key points are there. If we now go to a natural history study, because there's sometimes confusion about differences between a registry and a natural history study, and you'll be hearing about registry from Vanessa and registry and natural history study from Ashley. So they have very similar, they can be very similar, and they sort of overlap. This is taken from an FDA slide deck from an FDA officer, Ann Pariser. And basically, as this shows, a natural history study is a type of registry. It's a type of registry that's really trying to understand the disease prospectively. There are other types of registry studies. There's sort of regulators can require somebody to do, a sponsor or a company to do a post-marketing registry. So everybody who receives a treatment for a particular disease is enrolled in this registry study. They're not testing the intervention, but they're just observing what would happen over years with particular outcomes for patients that are given a particular treatment. But the natural history study is a different type of study. It's really trying to define the arc of the disease, what the disease is. And it's really important to have natural history studies, especially in ultra-rare diseases. And I think many of us who work in rare disease drug development really feel that natural history studies and registries are really critical to be able to develop and get approved therapies for rare and particular ultra-rare diseases. And I think the reason is because these diseases are so rare, we really don't know in a broad population what happens over time. I think you all have very intimate knowledge of the disease, but broadly speaking, you know, people who are developing drugs have, you know, try to get this knowledge but don't necessarily have it. Regulators also want to be able to understand how will you know if you give a drug to a patient with an ultra-rare disease and there's a change, how do you know that that change wouldn't have happened just by chance? We all think of diseases as sort of changing over time, and there's a pattern, but the reality is that things in different diseases can go up and down, and kind of like the stock market, there can be, you know, there's a pattern of the disease, but within that pattern, there are ups and downs. So if you do a short study, how do you know that these up and down changes that you see with your drug wouldn't have happened by chance? And a natural history study combined with registry allows us to really understand that. And with that, allows us to actually not do a placebo-controlled trial if we can avoid it. So with a very rare, rapidly progressive disease, obviously we would like to avoid giving patients and families, you know, no treatment, because that window that they're on placebo could be a critical period for being helped by the drug. So the natural history study provides a comparator for us to use in the absence of actually giving a placebo. And it's also serves a regulatory purposes. Regulators want adequate and well-controlled studies for registration, and that's in their official language. Studies need to be adequate and well-controlled. And if you don't do a placebo control, how are regulators or really anybody going to know what would have happened in the absence of the disease? You know, I think the data that Dr. Bankwitz presented on the AADC, where these children improved dramatically, that's an amazing case of improvement in function. And I think, you know, we can all see those videos and say, like, look, there's an improvement. But in many cases, we think that many therapies, especially when you're attacking the sort of root of the disease, are going to be most effective given very early and preventing a decline in the disease. And for that type of study, a natural history comparator is particularly important. And I just wanted to touch on one important point before handing over to Vanessa. What we hear a lot is, well, there's a lot of studies published in the literature about this disease. Why can't you just use these older papers? We know about this disease. And I think there is data published, but there's also bias in the published data. The severe cases tend to be reported. Standard of care for diseases change over time. So regulators and really anybody to really have faith that a treatment is having an effect, you need to compare it to a group of patients that are sort of studied of a similar population in a similar era. So not to compare patients in the 2018 timeframe with, you know, patients that were reported in 1960. And there was actually a study done, and this slide shows us, to really compare what kind of information do you get if you give a treatment and compare to a sort of active placebo control versus a historical control from many years before. So one study actually looked at a treatment for a particular condition. I won't go into the details. And people did several studies controlled at the same time or historical controlled. And they did not show the same result. The studies gave different results, and it's because the comparator group in the older studies functioned very differently. Their outcomes were much poorer than the outcomes for modern day comparator subjects. And so that made the treatment difference different. So it's really important not only to have good natural history and registry data, but it's also, you know, important to be able to have that so that you don't need to go back to potentially biased sources. So I'll stop now, and I'll hand over to Vanessa, who's going to talk about a patient registry that she's involved in.
Thanks, Adam. Mm-hmm. Yeah, that's it. All right. Oh, do I use this? Thank you. Okay. All right. Thank you, everybody. I'm Vanessa Ringel-Miller. I'm from Invitae. We are a genetics information company and a technology company that helps support the GM1 Foundation Patient Insights Network, or Patient Registry. So by show of hands, how many people knew that the Cure GM1 Foundation had a patient registry? Anybody? Hey, good. This is great. Okay, perfect. So what I'd like to do today is share with you some of the early learnings that we have from the program, and also describe for families who are not yet enrolled how they can join, and happy to take any questions. After today's sessions, I'm also available to help people check up on their accounts. So you can meet me outside. I can help you look at your accounts and make sure you're enrolled, or make sure your accounts are up to date. Okay, so what is a Patient Insights Network, what we call a PIN? This is our version of a patient registry. It's a centralized, comprehensive, and global database that allows patients to contribute their data, to do so in a way that protects their privacy, keeps their information, keeps their identifiable information safeguarded, and yet allows them to share their health experiences, their disease journey with the greater scientific community that's involved in GM1. At Invitae, we have a number of these programs. We actually host 85 patient registry programs for a variety of stakeholders, including advocacy organizations, including biopharmaceutical companies, including research centers, and the National Institutes of Health. So that's how we got involved. I started this work with our company back in 2007. We worked in many rare disease spaces, ultra-rare disease spaces, and continue our work today to engage stakeholders across all rare disease communities. This, I should point out too, this program has a few supporters seen by the logos on this slide. So I'd like to start by talking about the benefits of participation to patients and families, because at the core, we're a patient-centered resource. We really want to help people contribute their data, to learn from their data, and to provide information back that's meaningful. Families already have so much to deal with when diagnosed with a rare disease. And this is one way that we can help streamline the efforts of foundations and streamline the efforts of families to maximize their value. So families who participate in our program register an account, take online surveys, and contribute medical records type information. When they're taking surveys, families immediately have access to the data. They can see quickly and easily, and I'll show you examples, how their answers compare to others in the community, and it allows them to learn from each other. They can share that information with their healthcare provider, the doctors who are providing care. In fact, we have one good example from another rare disease community where a nonverbal child was experiencing pain, didn't know the source of the pain. The mother was very astute and noticed from the patient registry data that a large proportion of patients reported reflux. She had the pediatrician test the child was found to have reflux, treated, and the pain subsided. So it's just an example of how families can use this information to help better manage their health care. So in addition to helping improve understanding about the disease, it can also help improve access to information about research and clinical trials and relevant news developments. So through participation in the system, participants decide if they want to receive information from our team, from the coordinators who can share with them research-type communications. We send those to families. We put the information in their hands. They're in the driver's seat. They read about why they might qualify for such a study. They can read about what they can do as a next step, discuss the opportunity with their doctor, and decide if they want to learn more and contact a study recruiter. So there's a variety of attributes, and really what we want to do is ease the burden. You know, when living with a rare disease, families have so little that's under their control, and this is such a great way that they can make a difference directly. Let's see if I got it. Okay, so here's a quick glimpse at some of what we've accomplished, and you'll see a few more data charts too. So the program launched in November of 2017. We have participants from 14 countries. I should say this data is about a month old, a few weeks old, so the numbers are a little bit higher today. Most of the participants are from the US, but we do have participants from Europe, South America, even Australia. The participants, if you can see from the chart here, it might be kind of small font. Most are infantile onset, second most common are juvenile onset, then late infantile onset, and adult. One of the important things I should have mentioned when I talked about where people are coming from is that as of May of 2018, the platform is compliant with the European Union General Data Protection Regulation, or GDPR, because that's a mouthful. And that is really one of the privacy protections that's implemented across Europe, and now we're implementing across our systems to ensure people's information is safeguarded and protected according to publicized standards. And so with that, it really affords patients the most private system, but also keeps the information in their control. So a few more learnings across the community. One of the things we like to do is give back infographics to our foundation partners. I mentioned that we have many programs. We represent over 300 conditions, and so we develop infographics like what you see on the left here to help organizations promote the learnings from their community and raise the visibility and awareness of this disease. So in the patient registry to date, we have about a distribution of 43% female, 57% male, and maybe not surprising to most of you in the room here, 90% want to be contacted about research opportunities. That's a setting we let people decide on their own, and they can turn that off at any point. So here's an example of the homepage where you can log in and register your account. We have translated the site into 5 languages listed here, so Portuguese, Spanish, German, Taiwanese, and Japanese. From that page, if you haven't set up an account yet, it's a 2-step process. The first step, given the nature of this disorder, is one where the account holder, often a parent or guardian, will set up the account, enter your email address, so we have a way to contact you, set your username and password, and then on the second step, it's providing information about the participant who has a diagnosis, what their subtype is, what their date of birth is, so we know the age. We collect very limited address and phone number information. We don't collect that, again, trying to maintain people's privacy as much as possible. So once the account is set up, users have the ability to jump right into their first survey, and the surveys can be taken on mobile devices. This is a mobile responsive template. You can take it on a smartphone, on a tablet, on a laptop computer. Families are busy. You can stop and start surveys as you need to. If you get a telephone call in the middle, stop it, come and resume when you have time available. And we can send reminders to participants as well when their work is not yet completed. So as soon as participants have finished the survey, they have the ability to see the data, and that's one of the great givebacks and probably the most cited benefit of participating in these programs is participants see the data right from their accounts. So the first thing they see when they're looking at our View Data feature is the map that shows where people are located regionally. It doesn't display any address-specific information, but it gives a sense that you're not alone. Then participants can see the aggregate responses for the survey answers, and it compares your answer versus the rest of the community. You can also sort it and filter it by age or by subtype. There's some different filters you can apply to that. So a few other learnings to share with you, preliminary learnings. First, we looked at the diagnosis survey responses. We do survey people according to different topic areas: medical care, family history, diagnosis. We can add surveys as the needs arise. So we found that people are most commonly diagnosed by neurology or genetics. We found that about 72% of people report having genetic confirmation of their diagnosis. About 83% of participants noticed the signs and symptoms were noticed by family members, and that's what led them to the initial medical evaluation. And perhaps a story that's not uncommon among rare disease, about a third of participants, the diagnosis took 1 year or more from the times that the symptoms were first noticed. And that's something that Invitae really believes in, is how do we work on shortening that time to diagnosis to get people connected to therapies, to clinicians, to organizations and foundations sooner. Here's just a couple examples of some of the data to show you as well. We have a GM1 specific survey that participants take. We looked at the number of people who report diagnosis of a seizure disorder, so 10 participants with infantile onset, 8 participants with juvenile, 6 with late infantile onset. We asked participants to report on their ambulatory status. Across the subtypes, you can see about 15 walk with assistance or walk independently. And this is an interesting one. We also ask about impact and financial burden. And here we can see the estimated out-of-pocket annual expenses that families pay. And while, 9 report paying less than $1,000 annually, there are 3 who are paying between $15 and $50. And it could be more. You know, as we go and survey families, we start to really understand what the burden and cost of care is. So I think these are important numbers that really demonstrate the impact on families. So I always like to cover at the end kind of the important questions that come up. And one of the things that comes up is privacy and security. In our systems, patients own their data. They can opt in and out at any time. They can decide whether or not they share their de-identified information, and they can withdraw that information from our system. So it's really in their control, and they're in the driver's seat. And you can decide, as I mentioned earlier, whether you want to receive contacts about clinical trials and information. It's only from the coordinator. We don't share participant email addresses unless they have specifically requested that we share it. And we're pretty established in doing this. We started doing these programs, patient-centered, secure online programs in 2007. And our platform is really developed on standards that have been implemented from our work with the NIH. So our programs are managed according to a specific security standard called FISMA, which is an acronym, Federal Information Security Management Act, also another mouthful. But here the goal is just to ensure that the systems are handled and designed in as secure of a framework as possible. We have a separate data portal for investigators, for people who want to use the data, so not just investigators. Anyone who's not a participant and wants to see the aggregate data charts and use the data has the ability to do so through a dedicated data portal. So we believe in making this information available. As I mentioned at our table earlier, we believe that information is more powerful and more valuable when it's shared. And we know that families want to share their data in a way that protects their privacy. So lastly, we're growing. This program has really collected a lot of good preliminary information, and this is one of the important steps that families can take in order to bridge the gaps that exist today in knowledge, information, and access. So we can help families become connected, but we can also amplify the needs of families, really summarize those and put them out there, allow the foundation to put them out there, allow investigators to put them out there, and really call investigators to action by demonstrating organized and well-formed patient communities. So I'll end there quickly. I'm happy to take questions later on at the conclusion of our panel, but I did want to point out again, our website is listed at the bottom should you like to join. Please feel welcome to contact us. We have the connect@invitae.com email address. Our foundation contact is Dawn, so please feel welcome to reach out there as well. And again, if there are any participants who want to check up on their accounts, I'm happy to do that after the close of today's session. Thank you.
Thanks, Vanessa. And our next speaker is Ashley Winslow from the Penn Rare Disease Center.
So I wanted to go over real quick with the community. It's really important for us to get your feedback. So please feel free to give that directly to myself, Christine, Dawn, really anyone, kind of your thoughts on what we're going to present today. I will make sure if you want my email address. I don't remember if I have it on the last slide, but I'm happy to give you my business card so you can reach out. It's really important when we design our natural history studies that we get feedback from the community to understand, is this something that you'd be interested in participating in? Is it too burdensome? Is it relevant? Are the endpoints something that really are going to help us understand what your son or daughter is going through on a daily basis? So that's really important in everything we do. So I wanted to talk specifically about our natural history study. Now, Penn acts as a coordinating center, and what that means is we're driving a clinical development effort. We're trying to run a natural history study, but we may not be the actual site that your son or daughter is going to go to. We're actually at this phase developing the protocol to really pinpoint what endpoints and outcome measures that we want to focus on in our study. But we're going to be reaching out in the next phase to clinicians across the US, Canada, Brazil, the UK, Europe, to try to identify clinicians that have patients that would like to participate and have the ability to participate. So Penn is, again, the coordinating site, and we help really keep the efforts moving forward, but we are not the actual site necessarily that you would be participating through. So we work through a network of clinical advisors. We're not experts in the clinical presentation of GM1, but we work with advisors like Roberto, Giuliani, Janine Jarnes-Utz, and Lauren Clark, who Roberto's based in Brazil, and Janine and Lauren between Canada and the US, and then a series of other key opinion leaders that really represent kind of a global perspective on clinical care for GM1. And that's really important to us, is not just to understand maybe what state-of-the-art care is in the US or in the UK, or an expert out of Brazil, but really get a global picture of what GM1 looks like, how it's diagnosed in other countries, what access to care looks like. And sometimes that can really be a differentiating factor for genetic disorders, is even just getting access to that genetic sequencing. So this is just a timeline, very just broad overview. We've done an investigator and site identification through our key opinion leaders. And right now we're really in the protocol development phase, and we're actually very much nearing the end of that protocol development. So the aims of our natural history study are really important for us to be transparent and communicate to you as parents what we're aiming to do. So our number one aim is to develop a natural history comparator data set for GM1 clinical trials. And this kind of goes back to some of the discussions earlier. In a rare disease community that's quite small, you don't always have the opportunity, nor would you want to participate in a placebo-controlled study. So if we can generate robust clinical data and understanding of the progression of symptoms in infantile, juvenile, all spectrum of GM1 presentation, the hope is that we don't have to run a placebo arm. That becomes something that we don't do with gene therapy, and as Dr. Wilson talked about, that's something that we are aiming to do. But the hope is a data set that we generate that mirrors a clinical trial and that would be available to the community, not just for a gene therapy, but other biotech and pharma that are coming down the pipeline as well. And then the hopes are that when you move into a clinical trial, you have endpoints that are relevant, that measure change over time, and that are sensitive to change. And that can be hard to figure out for a rare disease ahead of time. So what we try to do is develop our natural history studies to test those ahead of time, to be able to answer the questions, are these sensitive, and do they show change over time? So it becomes, it's developing a data set, but also ensuring that our clinical study design is relevant when we move into the clinic. So this is where we'd like your feedback today or over the next few months. What we're hoping to do is to run an infantile and juvenile focused GM1 study. It'll be a two-part study, which I'll go into a little bit more detail in my next slides, but one will be in clinic for those that can access clinical sites, and we're hoping to distribute those regionally and not just at one site, so there'll be further access no matter where you are in the world. But there's going to be a lot of participants who can't necessarily get to that site for a number of reasons. So we'd like to launch a kind of mirrored web version of some of our questions. We hope this will be a 3-year study with 6-month interval between visits or between that web portal data update. And those visits in the clinic, we expect to be a one full day of testing. The primary overall assessments we're looking and questions we want to focus on are developmental milestones for your children, disease progression, understanding of motor function and language, and then also accessing plasma, urine, and CSF. We know that's a hard ask, getting access to CSF. We do usually make this optional because we know it affects recruitment, but whenever that's possible, we try to build it into our protocols because it can help us to interpret clinical results once we move forward in a clinical trial. So for that study number 1 that's in the clinic, our proposed inclusion criteria, and again, this is where we'd like to get a feedback from the community to understand if this makes sense. This is to incorporate both infantile and juvenile presentation. So we would have a minimum age for enrollment of 4 months of age. For infantile, what we've heard back from the clinical experts is that there should be documented symptom onset by 6 months of age with hypotonia, or that floppy kind of central muscle tone, on exam by a clinician or history by the parent or caregiver. And then for juvenile presentation, it's symptom onset usually after 2 years of age to include abnormal gait and or loss of vocabulary. So again, if this doesn't represent your infantile or juvenile or type 1, type 2 presentation of your children, please let us know because this becomes important for us to make sure that we're capturing the right set of features, symptoms when we design this study. And then we look for confirmation of disease through GLB1 documented mutations and reduced enzyme activity assays. And then other criteria we've had questions about, and this could also change, is understanding of what other medications your son or daughter might be on. Miglustat and Tanganil treatment is allowed. We don't see that as being a problem. Part of this is understanding also in a natural history study what the general population is normally on, so capturing medication use. And then feeding tube would also be allowed. The proposed outcome measures for the box really represents both infantile and juvenile, and then below the box under juvenile are domains that we're looking to get more clarity on how to measure these in the clinic for the juvenile population. But primarily, we're looking to mirror clinical trials, so survival is hugely important for us to understand and something we obviously would like to prolong, but also gain of function in vocabulary, motor development, And those are things that can be captured by the Bailey scale and Vineland. So these are clinical outcome measures that are normally used in neurodevelopmental disorders or neuromuscular disorders. Understanding feeding tube placement, that choice that both the parent and clinician makes, what time that happens, understanding what factors really are part of that decision process is important for us to understand that when we move into the clinic, is that something that's significantly representing that treatment is being productive and actually helping the outcomes and quality of life of your children. And then again, CSF biomarkers are important, but can be a big ask for the community. We will also look at peripheral changes and central changes through things like MRI and X-rays. So we know liver function, the size of the liver, spleen can be something that is very much present in very early on in infantile population. So we will likely track this over time as well. And then we're looking haven't yet quite decided how this would be executed, but electrophysical biomarkers, things like nerve conduction, other things that might be interesting to look at visual evoked potential, but these are a little bit more state-of-the-art and can sometimes be harder to execute in a multi-center study, but we'd like to keep that in mind. And then for the juvenile population, again, kind of looking at the overall domains that we've heard are really important for this population, dysphagia or swallowing function, gait function, motor, language, and respiratory function. So we're looking into those in more detail, and we'll be talking to clinicians over the coming months about exactly how to measure those accurately. So then our study number 2, this is for those who can't participate at a clinical site for various reasons. We'd still like to capture data that we hope the FDA or regulatory environment would be open to listening to and being receptive to, but this is a little bit of a new area where we're trying to push the boundaries. They're very much understanding and used to seeing the formal natural history study design, but things collected through web portals, that's a little bit more of a fuzzy gray area. We're going to try to push that conversation though. But importantly, regardless of whether the regulatory environment is open to it, we'd like to have a better understanding of kind of the global picture of GM1. So whether you can participate at a site, if you come from all corners of the world, we really hope to capture an understanding of some of the outcome measures that we think we can accurately measure in a web environment. Survival, feeding tube placement, seizure onset and frequency, and then Vineland, again, is a questionnaire-based kind of scale to look at milestone and motor and function development. So how will this data be used? So the data, we have our aims and there's a few ways they can be used, and we actually hope that really others besides ourselves use these data over time. But the data can be used to better understand natural progression of the disease, as well as to evaluate effectiveness of a new treatment, which is our goal really in initiating this study. We also need to know how that's affected by care, differences in standard of care in different regions of the world, because that can hugely affect outcomes differently in the US versus Europe versus Brazil. Natural history study data are often used when a clinical trial does not have a placebo arm. We kind of covered that in the context of gene therapy trials, but the hope is if this data set's made available, that that's also true for other future trials that may or may not be gene therapy. And then our data, one of our principles as a center is the data should never support just one investigational product, that these data should support other investigational products out there, so the data does become available to the community at large. And we do try to really push that qualified researchers drive further research insights off this data. So we really try to push it back into the community and get people working on it as well. So that is my email. Glad I put that up there. I wasn't sure if I did, but please feel free to reach out to me. Let me know if this is something that, if there's certain things when describing your son or daughter that didn't quite jive with our perspective and what we're hearing from clinicians. We'd like to know that early so that we don't design something that isn't going to fit with the community. You want me to grab Vanessa?
Vanessa, do you want to come up here too? So there was a question here. Okay.
I think I read this correctly on the slide that was talking about the patient, in order for a patient to be able to be part of your study. If it was infantile, symptoms had to show before 6 months, and if it was juvenile, they had to show after 4 years. What about late infantile, which comes in about 1 year? Is that something that you are going to be studying also or including in your history study?
So the criteria for infantile was before 6 months, and I think for juvenile we have after 2 years of age. So there is kind of an in-between area that we're trying to understand. Is that a nomenclature difference? Because we've heard from a few clinicians that they see this as that there's not necessarily a late infantile. They would really push either into infantile or juvenile. So for those that present right now in the middle of what we're classifying, that's something that we need to better understand how many people fall into that category and what truly that represents as far as progression. So I think largely when we go into our natural history studies, we try to have a more open, broad criteria to really get a fuller understanding of the spectrum of presentation, whereas a clinical trial may be more narrow. So that's something that if that's not going to fit your granddaughter's condition, we definitely would like to better understand.
Right, it doesn't fit hers because she was diagnosed with late juvenile and first symptoms occurred at about 1 year.
Okay.
So I'm not really sure that you would, that based on what I saw on the screen, that she would be able to be part of this, you know, if you wanted to find out information about her. I don't know where you'd put her.
Yeah, no, that's fair, and that's something that we really need to understand right now, because that's where we've been discussing with clinicians. There has been a little bit of a difference in classifying that late infantile population as either juvenile or as a truly distinct subset. We've heard differences of opinion, so that's very interesting to know, and I really would encourage anyone else who falls into a similar category to let us know. 2 patients here. Okay. So that's something we can take back to. We work in a really iterative environment with our key opinion leaders and clinicians. So this is why we think it's really important to talk to the community and take that back to them to say, look, this is what we're hearing. And this might be something that we would consider really changing that criteria. We haven't started our study, so there is really a lot of time that we can, I think something like that we could take back to the group and consider. So I appreciate that. And please reach out if anyone else feels similarly. We'd like to know.
Okay, my son, Chris, is 15 years old, and he's seen at the University of Minnesota on a yearly basis with Dr. Utz and Dr. Whitley, and through their clinical trial for Zaveska. And they're doing a natural history study on him. Is that separate from what you're talking about?
So we haven't started yet, but our next phase is reaching out to clinicians. We've worked with Janine already in kind of this design, and that's really working off of the natural history study that she's currently working on and in the past. We know NIH has a big effort that a lot of parents are involved in, so we will be reaching out to those clinicians to try to get the involvement going forward, merged, or also understanding if the data being collected is different. So what we're trying to do is a little bit different than what has been done in the past for clinical natural history understanding. We want to mirror a clinical trial in a very formal regulatory type environment so that the data can be directly compared. And sometimes in the past, if that data hasn't been collected in a similar way, they're not as directly comparable.
So I was wondering if we would want to sign up. Is this on?
I think so.
Okay. Would we want to sign up for your study as well as their study, or would that just be duplicating?
We will help you figure that out as we start to work with these clinicians.
Okay.
I think Jim had a comment.
Yeah, this is Jim Wilson. So I work with Ashley on this, and what we encounter time and time again, not only us but the community, is that as new product concepts come forward and are brought into the clinic, that the community has not set the stage in the infrastructure for natural history studies that can be used in open label studies as comparator groups. So they have to be GCP compliant, and they're essentially like a clinical protocol. So virtually 99% of the quote unquote natural history studies that are done based on foundation money. I tell you, NIH money. They're phenomenal efforts by committed communities and physicians, but then when you go to the FDA and you want to avoid placebo-controlled studies, they're not adequate. And you can't bully the FDA because it doesn't have to do with how committed the physician is or how complete the data are. It has to do with the quality controls of the systems. And so, for example, a 30-patient natural history study like this is about $5 million to do, which would be equivalent to a clinical trial. And so, I bet it's frustrating if you've spent the time working with others to try to acquire information. I'm sure it's been incredibly helpful to set the stage for making decisions, and Janine's been, I can tell you, when we know Chet well, it's been some of the best and most available data that we have, and it's informed everything that we've done so far. But going forward, if we're entering in the era of first-in-human novel therapeutics, that we need to sort of prospectively acquire data that is compliant with FDA standards. And we every time we make attempts to try to assimilate information that's already been acquired, that's helpful, but at the end of the day, there has to be a body of data that has undergone all the quality assurance and the information systems are compliant, you know, beyond that. So, yeah.
Dr. Wilson, if this study is going to inform your work, and earlier you talked to us about a target date of potentially a late 2019 trial, right? This is a 3-year study that's still very much in prelim phase.
No, we're gonna launch it really soon.
Fair enough.
The natural history study.
Early 2019, right? The best case you'd have, call it 9 months worth of data. Is that really enough to inform the current trial, or is this really more about long-term and future iterations of that trial?
So, we're late. We should have done this before. Guilty as charged. And the problem is we couldn't find $5 million to do this until there was a prospect for a therapeutic trial. I think we have enough information now that would inform the design of a phase 1-2 study. But what we need to do is, in order to determine whether it's efficacious or not, that has to be compared against GCP compliant data. So those are two different things. Informing the design versus allowing the FDA to make a decision whether it's effective. And I think we have enough information now, although definitely with infantile, I think we have enough information. In the attenuated case, it's a broader spectrum of disease, and that's something that I think would be more helpful, maybe necessary to inform the trial. But do you see those are two different things, and that will go ahead if we're confident enough about the endpoints, and you often acquire a lot of data as primary and secondary endpoints that may all not be necessary to convince the FDA for approval. And we're also talking about a phase 1-2 study. But I've never seen a disease that has established a full-on GCP-compliant prospective natural history study before the first-in-human study. It just never happens that way. What we're doing is we're just pushing as hard as we can to set the stage so that we don't delay it anymore. And then finally, I do want to make the point that that this is available to you, and this is available to whomever. You know, whoever needs this in order for them to make decisions about your trial or to conduct your trial, that is so key that it's available. Now, if industry wants availability, there may need to be a little bit of a cost because we have to pay for this ourselves, but it's not prohibitive, and you know there are ways to stratify it. But it is meant, and it's not a lead-in study. It's not a lead-in study for gene therapy. It's not. And that has to be clear. This is a natural history study that can be deployed against any one interventional study. It's a little challenging because we don't know how they're going to design their interventional studies. That's why it's sort of a catch-all of endpoints. But I would reinforce, the input that we can get here would be really useful, because once you launch and you start acquiring these data, you can pivot a bit, but not a whole lot.
Gotcha, fair
Does
enough,
that help?
thank you. And I guess quick follow-up would be similar to the woman in front's question earlier. So there's a number of families in the GM1 community that are already a part of the NIH natural history study.
Right.
And so I'm certainly not familiar with, the GMP protocols and such things, that you were talking about earlier. But I assume theirs probably doesn't meet that threshold if you're looking to replicate and do your own. So how should we think about, for those of us that are participants now, how should we think about doing both, switching to yours, things of that nature?
Well, I've reached out to Dr. Tiff to try to initiate a dialogue so we can be helpful, and I'm going to push as hard as I can. She's only 2-hour train ride away from me, so. So, look, this is the hardest thing we do. This is trying to align and pull this information together so that it doesn't duplicate efforts for you all, and we take it very seriously, and it usually doesn't go as well as we'd prefer, but we usually end up getting there. But definitely with respect to Minnesota, where Janine's the PI.
Yeah,
So, I mean, we went to Janine, and we're still trying to make sure that we get the connection and with NIH, and so I can promise you we're going to do our best and just to be as open as we can.
Just to clarify one point, I don't think what we're seeing is that the people are participating in the current natural history studies, that data can't be useful. I think that data could be useful. There are probably levels of, endpoints might be different. The way that the endpoints are assessed and collected might be more or less rigorous, but I don't think that going to that site and going every year is a waste of time. I think that any data you can collect over time is useful. There's a spectrum of useful data, and there's a spectrum of how regulators view it. But I don't think the message is that site A is useless, versus other sites. So I just want to provide that context.
Could I bring up another point, though? A heads up for you all. And this is my opinion. I work for the academy, so I have academic freedom, and I can say whatever I want. But let's be careful about lead-in studies, which means you enroll in a study to acquire the data that would be the baseline data for the interventional study. Let's be careful about this. Let's try to figure out a way to generate data that would obviate the need for those, because if every one of these had a lead-in study, you're running around enrolling your kids in all the lead-in studies. That's wasteful. And that's at this point we don't have it, but I got to tell you, San Filippo, right? There must be, I don't know how many. There are more trials than there are research subjects, but many of these are lead-in studies, and I see parents going from lead-in study, which is essential natural history study, to natural history study, just running themselves ragged to make sure that they could participate in the clinical trial. And that's something that I think can be avoided, and I would encourage us to try to avoid doing that. And if we can do this in a way in which the other 3 biotech companies, when they're ready, could have access to this, then they don't need it for the lead-in study. And that's
challenge question then.
Yes.
We have natural history studies out there. I understand that you may view some of their data as questionable on intake, but why would we spend $5 million on another natural history study when we could spend that $5 million working towards cures?
Well, first of all, it's not
and
me.
why would we participate in a natural history study when we already have natural history studies, when we could be using our money towards a cure?
It's not me. It's the regulators. And it's, the data have to be acquired in a manner in which they're compliant with certain basic principles of good clinical practices. And if they're not, they can't be used to determine whether a drug worked. It's not me. It's not me.
Okay, I understand that. So who are these regulators that we keep talking about that parents need to be talking to?
Well, I would be careful about that. I mean,
Okay.
you can talk to whatever regulators you want, but I understand the basic principles of data integrity. It's not bad data or whatever. And that's good clinical practices. And if something isn't acquired under good clinical practices, I'm not going to tell FDA that they have to change their basic standards of good clinical practices. The bottom line is, in order for us to get the $5 million to do this work, companies who have the resources to develop therapies want us to do it. It's not either or, and it's not philanthropic money. Companies that have the resources to develop products know this has to be done, and they don't blink at it. They don't blink at it. So we can get the $5 million to do this.
This is a standard part of clinical development. I used to work for Pfizer. I used to work in academia. I've seen both sides. This is a natural history study run in this format is just a different type of study. Oftentimes when a disease is new or in starting out, a natural history study answers the questions of what is symptom progression over time, what does standard care look like. It's more clinical, answering clinical questions. When you move into progressing a drug into the clinic, pharma and biotech, especially in rare disease, often have to collect a comparator data set in answering different questions and format it in a different way. And it just becomes a cost. While the study is separate from clinical trials, we're actually doing that to try to get ahead of the clinical trial. And that's something that hasn't been done in the past. Usually it's catch-up. There's a number of companies in the past 9 months that have gotten delayed. They got to the finish line, they showed efficacy, they went to the FDA, tried to get it approved, and FDA said, hold on, you don't have a natural history study. The data you're showing us was collected for a different purpose to answer clinical questions. It's not up to our standards. So what we're trying to do is change that paradigm and get ahead of that. And unfortunately, it does cost a lot of money. It's the cost of developing a drug. We're trying to drive down that cost by, yes, the first one's going to be $5 million, but we're trying to now share that with the community so not everyone behind us has to spend $5 million. So it sounds like a lot up front, but the hope is that that drives down costs over time. And NIH's and Minnesota's efforts have been really key for us trying to plan our study. It's been our foundation all along, and that's how you want it to proceed, but it's an evolution, and it progresses. It's just an unfortunately long and costly process.
So the information that the NIH has collected is not something you can use. You have to have independent natural history studies. And my second part of that question is, since my granddaughter has already participated in the NIH study, is there any point in her participating in another organization's natural history study?
So the data that's been collected to date from other studies has been hugely helpful. I'll reiterate that, absolutely foundational for what we're planning. So there's absolute value that's already provided hand over fist for the community, and that's huge for you to be able to contribute that. That is not useless data. It's allowed us to move into a disease that we think we understand what a clinical trial would look like. And that becomes a big point of a decision for pharma, biotech to move into a space is, do we even have a clinical endpoint that we can test against? So that data from Minnesota, NIH, is very powerful in that kind of foundational question. As far as continued participation, we'll be reaching out to these investigators, trying to understand where we might help, where we might get them to participate in our study, where we could help in their efforts. We'd really try to be collaborative, but at the end of the day, there may be a point where you decide whether to continue your son or daughter in both studies or move over to one. And we will be transparent in our efforts and what that looks like. We deal with this in a lot of communities where there's a lot of questions about the efforts to date and what we're trying to do. But it's not a waste of your time to be a part of those studies. We're just trying to kind of move into the next phase that's really focused on getting a drug approved. So we will communicate with you, we'll communicate with Christine and Cure GM1 to try to answer your questions along the way.
But we wouldn't discourage anyone from continuing to do what you're doing. You know, we just need to accrue enough subjects to get statistical significance so that we can power an interventional study. And we're going ex-US as well, so we're going international. So I don't think anyone should feel that it's necessary for anyone in this room, really, to participate in our study. In contrast to the NIH, you know, we're here to share with you what we're trying to do, and we're going to aggressively try to enroll subjects in this in the full spectrum of disease, so it's powered enough. But once it's powered enough, then you don't have your individual participation in no way, shape, or form should influence whether you would qualify for any clinical trials anywhere, but surely nothing that we're involved in.
That's a good point. Gori?
I just wanted to follow up on what the two grandmothers in the second row were saying. I think with GM1 gangliosidosis, it's a continuous spectrum of phenotypes, and there are a lot of infantile patients, and there's also a lot of juvenile patients, but there is this important patient population in the middle, the late infantile group. And the problem with the infantiles is that their disease progression is too rapid sometimes to develop a good clinical trial. And with the juveniles, their progression can be very slow. So I think for pharma, they're looking for a group of patients where the progression isn't too fast or too slow, so that you have time to come in for an intervention. So with your criteria at the moment, you do run the risk of really excluding this really important population in the middle, the late infantile group, or whatever we want to call them. They're not quite infantile and they're not quite late juvenile. They're in the middle. They're late infantile.
I absolutely appreciate that. That's what we're trying to better understand, so that's something we'll take back to our clinicians and discuss in further detail.
I want to add something to what was said by Dr. Wilson. You have to understand, as parents, that if a company wants to develop a new therapy, it's an expensive endeavor. We have to raise money, a lot of money. We have to raise, usually to begin the clinical development, $20 million, $50 million. The people that fund us are very smart people. If there are no standards which we can go against, we will not get the funding, so the therapy will not be developed. So what is absolutely critical is that these standards are set. And for a disease like GM1, apparently the standards that we are used to, goodwill, effort by doctors and so on, a little disappointing by the NIH, to be honest with you, because they should have followed the CGP guidelines, in my opinion, but anyway, we have to set these standards. The standards are going to be useful to compare our results with these standards which will be set with this clinical trial. And it's an essential element for us to be able to succeed in raising the money and developing the therapies. It's the way it is. And I am very grateful for the effort that Penn University is doing, because somebody should have done it, and they decided to do it. And not only they decided to do it, but they will follow up, or concomitantly, almost they will try a gene therapy, which we all know it's an amazing opportunity for all the patients. At the end of the day, it's going to be helpful for everybody, including my own companies and all other companies that will enter this area of effort. And as we know, the future of every therapy is going to be combination. So we will be happy to combine our chaperones with the gene therapy. Enzyme replacement therapy will be combinable with chaperones and gene therapy. So the future, what we want for your kids, we want the best. The most of options and the best combinations are going to be useful only if we can get them to be registered. And in order to be registered, we need solid foundation for comparative data in which we can build our clinical programs.
So I wanted to make a plug if there's any other questions, for what Vanessa's doing with Invitae. We've talked to Invitae a few times. Vanessa and I have met, and a lot of what we try to think about is logistics of identifying patients all over the world, where they are, trying to get them enrolled, understand interest, identify clinical sites. That's a heavy lift. It's just that recruitment piece. We often try to collaborate with groups that have collected that data early. So if you're not registered yet and you're interested, I'd encourage you to register with Invitae because we're hoping to collaborate with them to try to accelerate our efforts too.